good afternoon. I think good morning also in the States. I'm really happy and glad to be here and to present, let's say, a novel and hot topic such as biostimulants, and in particular how these compounds and microbial inoculant can increase and improve horticultural and agronomic crop performance in a sustainable way. Well, the outline of my presentation, I will start with a brief introduction about the main challenges of the horticulture sector and the concept of sustainable agriculture, with a brief overview on plant biostimulants. Then I will show several case studies dealing with greenhouse and open field crops on how biostimulants they can improve economic profitability, but also the sustainability of horticultural crops. And in the second part of the presentation, I will talk about the implications of microbial and non-microbial biostimulants to enhance the product quality, so nutrient use efficiency, in particular nitrogen use efficiency, and also the tolerance to abiotic stresses. And the last couple of slides will be dedicated to the main conclusions, the take-home messages, and what are the future perspective in the sector of biostimulants. Okay, in the coming years, farmers and growers will have to deal with growing crops under suboptimal conditions. So low precipitation, high air temperature, water scarcity due to drought, all this dictated by the climate change. And also growers have to minimize the excessive exploitation of natural resources, in particular water. The modern horticulture is also facing other important challenges, such as the loss of soil fertility, and we have the big problem of contamination due to heavy metals, mineral oils, and unfortunately, all these contaminants will end up in our plates, in fruits and vegetables that we consume. So, what is the global scenario right now? The world horticulture system is facing several challenges and constraints, such as reduced availability of natural resources, as we said, the water, more rigorous environmental policies, low product selling price due to competition and especially globalization. And the consumer is becoming more aware. Consumer expectation is shifting towards functional, nutritional, and also environmentally product. For this reason, the scientific community, the scientists, the extension specialists, growers, producers, and even technicians should focus on finding tools and agricultural practices, sustainable agriculture practices, which are able to improve the crop productivity and the quality of our produce by reducing the production cost and by increasing the efficient use of resources, in particular water use efficiency and nutrient use efficiency, and to reduce the environmental footprint. It is well known that the golden rule of sustainable agriculture is that we must meet the needs of the present without compromising the ability of the future generations to meet their own needs. So, in few words, sustainable agriculture integrates three main goals: the environmental health, economic benefits, and quality of life, which is the social equity. In the frame of the farm-to-fork strategy in the European Union for a European circular economy, the EU aims by 2030 to reach the following targets for a sustainable food production. a reduction of the chemical pesticide by 50%, to reduce the nutrient losses by 50%, in order to reduce 20% of the fertilizer and increase the nitrogen, the nutrient use efficiency, in particular nitrogen. And a third target, and very important target, is to increase the percentage of organically farmed land by 25% within 2030 in the European Union countries. Several possibilities for traditional and innovative intervention to improve the performance of horticultural production in a sustainable way can be represented by intervention at the genetic level, at the climate control level, especially when we are dealing with greenhouse crop, post-harvest management. And during my talk, I will focus basically on agricultural practices, in particular biostimulants. As we know, we have several agricultural practices like a smart management and scheduling of irrigation, management of fertilization, organic matter management, solarization, and one of the most tricky and trendy agricultural practices used in the last 10-15 years is the use of plant biostimulants. Recently, the European Union decided to reshape the existing fertilizers regulation. Under this new regulation, plant biostimulants are defined by agricultural claims, so by claims of agricultural function. Yet, it's very important, this concept, their primary function is not to supply nutrients like fertilizer, neither to protect plant from pathogens and pests, so they are not considered PPP, plant protection products. And these EU fertilizing products are able to stimulate the nutrition process independently of the product nutrient content, with the sole aim to improve one of the following or more of the following characteristics of the plant, also plant rhizosphere, such as increasing nutrient use efficiency, abiotic stress tolerance, improving and modulating the quality traits, and also the availability of confined nutrients in the soil or the rhizosphere. Based on the same regulation, 2019/1009, the EU biostimulants are divided in two major groups, let's say like this. Non-microbial biostimulants, humic substances, protein hydrolysate coming from plant or animal sources, seaweed extracts, and also an important category, which are the microbial inoculants, the microbial biostimulants, in particular the endophytic fungi, mycorrhiza, and plant promoting growth bacteria such as Azotobacter, Azotospirillum, and Rhizobium. So as I have reported in the outline of my presentation, biostimulants should be considered as a tool to improve the economic profitability of the growers. What are the different ways to increase the farmer income? So biostimulant could increase the marketable production per unit area, an additional kilogram per square meter when we are talking about greenhouse crops, or additional tons per hectare when we are dealing with open field crops. Biostimulant could be a good solution to increase the selling price of the product by increasing the quality traits and characteristics of this product, and most importantly, by decreasing the production cost. As we said before, decreasing the production cost is decreasing the quantity of chemical fertilizer that we are using. So biostimulants can increase crop productivity. This is the first case study coming from central Italy on greenhouse fresh tomato, where 3 biostimulants were tested: a tropical plant extract, a legume-derived protein hydrolysate, and a seaweed extract. let's pay attention to the number of foliar applications that were applied during the whole growing cycle. 4 treatments were applied at 10 days interval, and the first application, foliar application, started at the first fruit set phenological stage. As we can see in the figure, we can observe that seaweed extract and legume-derived protein hydrolysate were able to increase and to boost the marketable yield by 7%, with the highest value reached with tropical plant extract with 11% of marketable yield increase compared to control treatment or untreated plants. However, it is very important to keep in mind that the biostimulant action of protein hydrolysate can vary depending on the species and also on the dose of application. For this reason, we conducted another experiment, this time in South Italy, under greenhouse experiment, where 2 concentrations of plant-derived protein hydrolysate were adopted, at 2.5 milliliter per liter and 5 milliliter per liter, but the foliar application was more frequently. We passed it from 4 times to 9 times, and we started directly after transplanting, 15 days after transplanting, and the interval was always 10-day interval. Here we can observe from this figure how the application of the legume-derived protein hydrolysate at a dose of 2.5 milliliter and 5 milliliter per liter increased the marketable yield in both cultivar by 12 and 21 degrees respectively. So in this slide, the partial budget analysis was performed based on these 2 experiments on greenhouse fresh tomato in order to evaluate the economic advantage or the economic benefits that may accrue to grower by applying these biostimulants. As we can see, for example, in the 4th column here, the added marketable yields due to biostimulants, this 11% increase, 6, 7%, and 21% when it was doubled in comparison to the control. The positive effect associated with biostimulant treated plant involved an added gross return which ranged between 4,000 and 7,000 euro per hectare, reaching 16,000 in the case of the double dose. As we can see, among the total variable costs, we can notice the cost due to harvest, post-harvest, the cost due to the biostimulant product, and for the foliar spraying. At the end of this partial budget analysis, we were able to determine the added net return, which is the differences between the added gross return due to the increase in the commercial production compared to the control, minus the total added variable costs, the variable cost due to harvest, post-harvest, spraying, and biostimulants. And we can notice how the added net return ranged between 2,000 euro and 5,000 euro for the greenhouse tomato. When we compare these results with other scientific findings about extensive crops and grapevine, for example, wheat, grapevine, and processing tomato, basically crops growing under open field conditions, we can notice that the added net return was relatively low compared to the added net returns that we observe for tomato under greenhouse conditions. So the highest added net returns that we observed under protected cultivation in comparison to the open field crop could be due to 2 reasons. The first one, under greenhouse condition, we have a higher frequency in the biostimulant application, while in open field crop, due to cost, we have a low biostimulant application, 1, 2, maximum 3. And another reason, very interesting, is the following: that under greenhouse condition, we have a favorable environmental conditions. We have a higher relative humidity, leading to an increase in the permeability and the uptake of this biostimulant product, leading to an increase in the commercial yield. In addition to the economic profitability of biostimulants could also be considered a tool, an efficient tool for improving the environmental sustainability. In fact, through the life cycle assessment, which covered all the processes related to crop cultivation, from raw material cultivation to harvest, we are able to quantify the kilogram of carbon dioxide equivalent per tons of crop produced. Basically, the life cycle assessment achieves the following objective: to assess the overall environmental impact of crop production chain and to compare different ways, in our case, biostimulants versus non-biostimulants, managing the production chain. The experiment was carried out under protected cultivation greenhouse crop using spinach as tested crop. The experimental design was so simple. We had two combinations of biostimulant: a controlled treatment without any application of biostimulant, and a biostimulant product coming from legume-derived protein hydrolysate. And we have 4 fertilization regime: an unfertilized regime, and 15, 30, and 45 kilograms of nitrogen per hectare. In this figure, we can observe the total carbon footprint of harvest spinach. The results can reveal that the CO2 equivalent emissions of the greenhouse spinach were reduced due to the biostimulant application. As we can see here, the highest carbon emission reduction, this 25%, was related to the unfertilized treatment together with the application of the legume-derived protein hydrolysate. While this reduction was very limited, it was about 7% with the application of the biostimulants, when the nitrogen fertilization was 45 kilograms of nitrogen per hectare. So we can conclude that the plant biostimulants have the potential to reduce the greenhouse gas emissions of vegetable production under greenhouse conditions. Another important take-home message is that the label of carbon footprint is certainly a distinctive element and an indication of the quality improvement of the product. We can talk about a green product. Another aspect, a positive aspect of biostimulants, is the implication of biostimulants for improving and enhancing the produce quality. As we know, the term quality is a complex term. Recently, the quality of fruit and vegetables was coined by a couple of researchers who defined that the quality of fruits and vegetables constitute a dynamic composite of their physicochemical properties and consumer perception. They include both intrinsic characteristics dictated by genotypic, agro-environmental, and post-harvest factor, and also extrinsic characteristics influenced by the socio-economic and marketing factor. However, in the last years, consumer, nutritionists, and even the scientific community is much more interested with the functional quality of our produce. Functional quality is linked to potentially bioactive phytochemical compounds like glucosinolate in the brassica, lycopene in tomato, carotenoids in carrots or food, a concept spurred by consumer interest in healthy eating combined with gastronomic delight. Here we can see an experiment carried out on tomato where we were able to demonstrate greenhouse tomato that foliar application of protein hydrolysate and tropical plant extract differently modulate fruit quality. As you can observe here, we did not observe any significant difference when the tropical plant extract was applied to the tomato plants, since no significant difference in lycopene content was observed between treated and untreated plant. However, we can observe that by applying protein hydrolysate, we observe 55% increase in the lycopene concentration. So the production and accumulation of this important antioxidant molecule with the protein hydrolysate application could be considered an extra value to support human health. Always in tomato, we was able also to observe that protein hydrolysate elicit dose-dependent increase in tomato, passing from 2.5 milliliter to 5 milliliter per liter. We observe an increasing in lycopene concentration between 14 and up to 35%. An explanation of this accumulation was associated to the changes in potassium uptake and concentration in leaves and fruits, which may have influenced the accumulation of this antioxidant in fruits, since it is well known from the scientific literature that potassium can be involved in the process of carotenoid biosynthesis. Not only non-microbial biostimulants like tropical plant extracts and legume-derived protein hydrolysate, also important endophytic fungi like mycorrhiza, which are able to establish symbiotic relationship with the roots of most of the agricultural crops, with few exceptions, some plants coming from the Brassica and Chenopodiaceae families. For instance, arbuscular mycorrhiza fungi are able to develop a network of external hyphae capable of extending the surface area up to 40 times, which lead to a higher nutrient uptake and assimilation of key macro and micro elements. And arbuscular mycorrhiza can also modify host plant primary and especially secondary metabolism, which can involve key enzymes and lead to the accumulation of these famous secondary metabolites or phytochemicals. In this experiment, which was carried out in the frame of a national project on globe artichoke, one of the most important horticultural crops in open field in Italy, and it is also cultivated in several European countries, Spain, North Africa, but also in California, Argentina. In this experiment, two seed propagated cultivars were used, Istar and Dromolo, and the seeds of these two cultivars were inoculated before transplanting with a consortium of two endophytic fungi, Trichoderma and mycorrhiza, and we were able at the end of the experiment, based on analysis carried out on globe artichoke head, to observe an increase in the ABTS, the antioxidant activity, and especially in total phenolics of coated globe artichoke. Probably the arbuscular mycorrhiza fungi can affect the secondary metabolism of colonized roots by enhancing the antioxidant enzymatic system's activity. Also in leafy vegetables, in this case, in particular in red oak leaf lettuce, cultivated also under open field conditions, we observed that arbuscular mycorrhiza fungi treated lettuce boosted the antioxidant activity and the total phenols. But the most interesting result was that the results indicated that the different strains of mycorrhiza, which were used in this experiment, modulate differently the concentration of this important polyphenol anthocyanin, where Rhizoglomus irregularis strain shows stronger ability in affecting plant metabolism in terms of these important antioxidant molecules. In addition to phytochemicals, also minerals play a crucial role in the human diet, since minerals can avoid several nutritional disorders and disease symptoms due to their well-known potential and functionality in the body metabolism. Based on a couple of studies carried out in the US and in Finland, it was demonstrated that the total dietary intake from fruit and vegetables for potassium, calcium, magnesium, and phosphorus was around 35%, 10%, 25%, and 15%. In this experiment, always on tomato, this time on tomato, but under greenhouse tomato, we was able to observe that tomato produced by arbuscular mycorrhizal fungi were able not only to increase the lycopene concentration, but they were able to increase macro and micronutrient concentration in the fruit tissue, in particular calcium, potassium, and phosphorus, and zinc by 27%, confirming that the use of arbuscular mycorrhizal fungi at transplanting could be considered an efficient agronomical biofortification tool. However, future research should focus on the selection of the best combination. That we should focus on a personalized combination between the species that we want to test or the cultivar, the most suitable strains, and the environmental condition or the agricultural practices, in order to increase these secondary metabolites, also known as phytochemicals, with health promoting activity, to be utilized as sustainable biotechnological tools for the production of safe and healthy plant foods. Similarly to the data that we observed on tomato, we conducted another greenhouse experiment, this time on zucchini squash, where before transplanting or during transplanting, we inoculated plant with a mycorrhiza, Rhizophagus irregulare and Funiliformis mossea, and we observed at the end of the experiment that the biofortification, the concentration of macro and micro element increased in mycorrhizal and non-mycorrhizal plant by 20 and 15% for the macronutrients, and by 14 and 17% for the micronutrients. Another important claim of biostimulants in the plant nutrition, the ability to enhance the nutrient availability, to improve the nutrient uptake and the nutrient assimilation. So it is very important, as we said before, when I talk about the farm-to-fork strategy, nitrogen use efficiency must be improved for two reasons, both for economic, for the growers, and environmental reasons. So biostimulants have the capacity to improve the nitrogen use efficiency, and this is one of the claims supporting their placing on the market of plant biostimulants. For this reason, in this experiment, the enhanced nutritional status that we observed in a greenhouse experiment conducted on tomato has often been associated with the ability of protein hydrolysate to modify the growth and morphology, the root system architecture of the root system, facilitating the nutrient uptake, in this case phosphorus, potassium, and magnesium, as a consequence of the increased root absorbing area. So a possible mechanism involved in the increased root hair numbers and lengths could be related to the stimulation of auxin production by the soluble peptides present in the plant-derived protein hydrolysate that we use. Another experiment conducted this time on baby spinach. The growing season, it was a winter-spring growing season. The biostimulant tested was once again the legume-derived protein hydrolysate. 4 treatments were applied during the whole growing cycle at weekly interval. And 4 nitrogen fertilization regimes were compared, unfertilized, 15, 30, and 45 kilograms of nitrogen per hectare. As we can observe that the foliar spray with protein hydrolysate were able to boost the fresh biomass by 33 and 24%, especially under suboptimal and optimal conditions, while this positive effect was very limited under luxury nitrogen condition. The same trend was observed for the nitrogen use efficiency, where under suboptimal and optimal conditions, the nitrogen use efficiency was around 16 and 25%, which is excellent, while under luxury nitrogen condition, the nitrogen use efficiency increase was one more time very limited and non-significant, with an increase of 8%. The implication of biostimulants for improving nitrogen use efficiency was also done using processing tomato as tested crop, but this time using a biopolymer-based biostimulants, which contain micronutrients, lignosulfonates, and root hair promoting peptides. 4 treatments were compared. We have the biopolymer-based biostimulant and a control treatment with 2 fertilizer regimes, a conventional and low input. And one more time, this is very important to highlight this issue, that biostimulants perform better when we have a suboptimal condition. In this time, we can observe that the marketable yield increased by 19% when the low input fertilizer regime was adopted, while under conventional fertilizer regime, the positive effect was still relevant, but as only at 10%. Now, concerning the third claim, and from my point of view, it's one of the most important claims. It's about biostimulant and tolerance to abiotic stress. As you know, farmers around the world only harvest on average 50% of the potential yield, because under open field conditions, plants undergo several types of pressure, biotic and abiotic stress. In the last 5 to 10 years, the scientific community and also the private company is focusing on designing and formulating a new generation of biostimulants, let's say functional biostimulants, which can resolve specific problems. Why? Because under open field conditions, we have environmental stress, which can cause oxidative stress with the production of reactive oxygen species. So these new types of biostimulants should have specific targets. So the main biostimulants target, which will be able to contrast the abiotic stress, should contain substances which are able to improve the stomatal conductance, the water use efficiency, the root to shoot ratio, which can able to trigger the key enzymes involved in nitrogen carbon metabolism, can increase antioxidant enzyme activity, increase osmolyte like proline content, etc. In the first experiment, in the first case study coming from a greenhouse trial on zucchini squash, we were able to observe that arbuscular mycorrhiza inoculation at transplanting was able to increase the salinity tolerance of greenhouse zucchini squash. The treatments were quite simple. two nutrient solution, a saline and a non-saline nutrient solution, two combination of microbial biostimulants, a mycorrhiza and non-mycorrhiza biostimulants, and two concentration of phosphorus, a low and a normal concentration of phosphorus. As we can observe in the first figure, we can see in all treatments, the saline water decrease the marketable fresh yield of zucchini squash in all treatments. However, the reduction in the mycorrhiza treated plant was much more lower than non-mycorrhiza plants. As you can observe, a reduction only in 8% versus 26% and 27 versus 43%. In the same experiment, we was able to measure also the chlorophyll content of leaves. We observed, for instance, that arbuscular mycorrhiza plant under saline condition had greener leaves, suggesting that salt interfered in chlorophyll synthesis more in non-mycorrhizal plant, as you can observe in this picture, than in mycorrhiza plants. In addition, the sodium concentration in zucchini leaves was lower in mycorrhiza than non-mycorrhiza plant grown under saline condition. This may indicate that sodium might be retained in the arbuscular mycorrhiza fungal hyphae. So the mycorrhiza can play a biofilter or can inhibit the translocation of this toxic cation to the shoot. Another possible mechanism, suggested mechanism, could be the lower sodium concentration in the arbuscular mycorrhiza plant compared to non-mycorrhiza plant might be explained by the dilution effect due to the growth enhancement by arbuscular mycorrhiza fungi plants. Also, protein hydrolysate application could be a possible tool to increase salinity tolerance of lettuce. As you know, lettuce, contrarily to zucchini squash, is considered highly sensitive to salinity. As we can see, 6 treatments were compared. We had 2 nutrient solutions, a saline and non-saline nutrient solution, and 3 biostimulants treatment, a control, a substrate drench with protein hydrolysate, and a combined application substrate drench and foliar application. As we can observe, the saline treatment decreased the shoot fresh weight in all treatments. But one more time, the application of protein hydrolysate, especially through substrate drench and foliar spray, decreases the shoot fresh weight only by 11% compared to the 33% in uncontrolled plants. An explanation of that could be the data reported in this table. In fact, we observed that the substrate drench application and foliar application with protein hydrolysate was able to alter the morphology of the lettuce root system, yielding a more extensive, let's say, absorbing area, which may be considered a mechanism of tolerance. Also, from a physiological point of view, we were able to observe that the protein hydrolysate treated plant had a better functioning photosynthetic apparatus. If we have a close look to the maximum quantum use efficiency of the photosystem 2, we can observe how the control plants had a reduction of 20% in comparison to plants coming from protein hydrolysate treated plant, indicating that the protein hydrolysate biostimulant on root or on leaf and root can delay the photoinhibition under salt stress. In the same experiment, thanks to the use of on-target metabolomics, we was able to understand the mode of action of this protein hydrolysate. We observed that the application of the biostimulants has increased the flavonoids, which play an important role in the scavenging of the excess production of the reactive oxygen species, the ROS. We observe also in the same experiment an accumulation of osmolyte, in particular proline, which play a positive role in osmotic adjustment. And we observe also an increase in upregulation of sterols, which regulates the stability and permeability of the cell membrane. As I said during my introduction, another important category of non-microbial plant biostimulants are the seaweed extract. As you know, we have a lot of commercial products on the market, going from brown microalgae, red microalgae, yellow microalgae. In this case, we used for a greenhouse experiment on zucchini squash, the brown seaweed extract, Eclonia maxima, which was applied 5 times foliarly during the whole growing season. The experimental design was quite simple: saline and non-saline nutrient solution with 2 biostimulant treatments, a control treatment, and a treatment with biostimulant coming from seaweed extracts. We can observe in this case that irrespective of the nutrient solution, which mean in saline and non-saline nutrient solution, we observe an increase in marketable yield of zucchini squash by 12% at the end of the experiment. The interesting output of this experiment that was observed from a leaf anatomical point of view. We observed that the size of the stomata was influenced by the seaweed extract application. As we can see, we observe lower guard cell in length and width in plants treated with seaweed extract. And for people who know physiology, smaller stomata or smaller guard cell command faster. They have a faster response for opening and closing stomata. So they have the possibility to sustain a higher photosynthetic rate, a higher water use efficiency, and probably this could explain the highest performance of 12% observed in seaweed extract. In the last case study concerning the implication of biostimulants and tolerance to abiotic stress, I will talk about the last generation of plant biostimulants, which are the combination of microbial and non-microbial biostimulants, because as you know, in terms of efficacy, there are 3 types of interaction. We have implicating between microbial and non-microbial biostimulants. We have the antagonistic, which occur in some cases with 2 microbial biostimulants, the additive, and the most important and tricky one, the synergistic effect of biostimulants. In this experiment, always under greenhouse condition, the tested crop was lettuce. We compared 6 treatments, 3 biostimulants treatments, no application, untreated, a tablet containing 2 endophytic fungi, mycorrhiza and trichoderma, and a third treatment, which is the tablet containing microbial plus foliar application of protein hydrolysate, which is the combined treatment, using a saline and a non-saline nutrient solution. We can observe from this figure, in particular the blue color, how the application of the tablet, which means the microbial biostimulant, was able to increase the performance of lettuce under both standard and saline nutrient solution by 28%. And this increase was higher when the combination of microbial and non-microbial plant biostimulants was used. So great accumulations that we can observe here for potassium and the reduced sodium concentration in the microbial treated lettuce may have helped the plant toward maintaining the osmotic potential in their cell, and also preventing accumulation of cellular sodium to toxic concentration. This agronomical performance that we observed on lettuce plant coming from the tablet or the combined treatment tablet, so microbial and non-microbial, were also validated at biochemical levels, where we measure several osmolyte and 2 antioxidant enzyme activity. As you know, for example, proline play an important role in protecting cell membranes and protein from damage. Here we can see how the tablet plus protein hydrolysate treated plant showed maximum, this A49, maximum accumulation of proline as compared to the other 2 treatment. Also, the peroxidase and catalase responded differently under different biostimulant application. We can see how peroxidase and especially catalase, which is the major enzyme for intracellular hydrogen peroxide degradation, was higher, as you can see here, in the biostimulant treated than untreated plants. So, some take-home messages. What are the main conclusions based on this several case study that I have presented earlier? Biostimulants have a great potential to improve crop performance, especially under environmental stress conditions, as we observed earlier, in case of abiotic stress and especially in case of low nitrogen input, as well as improving resource use efficiency, in particular nutrient and nitrogen use efficiency, and in some case there is an improve in the quality of the product. Plant biostimulants are defined, but what they do, the claims, but we still largely ignore how they do, the mechanism or the mode of action. Why? Because biostimulants products are substances with multiple bioactive compounds. We are not talking about a single molecule. The use of omic science, such as in the case of lettuce trial, greenhouse lettuce trial with protein hydrolysate and salinity, is essential to elucidate the molecular and physiological mechanisms. Elucidating the best combinations, this is very important. We cannot say that this biostimulant is better than that. We should select the best combination. Biostimulant, plant species, and environmental interaction is crucial in order to select optimal combination in terms of yield and in some case quality, and also optimizing application parameter. We should have a webinar in this sense. It's very important. Biostimulants should be applied like we show earlier, as primers directly after transplanting, or as rescuers during the stress, or as recovery or restorers once the stress or the abiotic stress is evident. The mode of action, foliar application, seed coating, by drench, by fertigation, and the rate of application. This is very important. And at which plant development stage. The synergistic effect, as we saw, combining microbial and non-microbial biostimulants, should constitute the core of future research aiming to design, to formulate, and to develop efficient plant biostimulant products with specific biostimulatory properties in terms of yield and nutrient use efficiency, in particular nitrogens and phosphorus. Finally, the use of environmental impact model, the life cycle assessment, the experiment about greenhouse spinach and the life cycle assessment was the first ever published paper on this topic. It's very important to have more data in order to validate this data, as well as economic analysis. The partial budget analysis are really necessary to assess whether The use of these biostimulants, microbial and non-microbial, can be considered really an environmentally friendly tool, able to provide satisfactory income or additional income for the farmer. Finally, I would like to present in 2019 with my colleague, Professor Giuseppe Colla, we launched the first book, it's in Italian language, dealing with the different category of biostimulants and the implication of biostimulants in horticulture, in floriculture, arboriculture, and in 2020, with the collaboration with Patrick Dujardin, Patrick Brown, Stefania Di Pascale, and once again Giuseppe Colla, we launched the first book about biostimulants, entitled Biostimulants for Sustainable Crop Production. This book was divided in three main parts: an introduction and biostimulant characterization. In the second part, we can see all the categories of biostimulants, non-microbial, such as humic substances, seaweed extracts, protein hydrolysate, silicon, and microbial, plant growth promoting rhizobacteria, arbuscular mycorrhiza fungi. And the third part deal with innovation and practical application, a very, very nice part with a lot of nice chapters, such as designing and formulating microbial and non-microbial biostimulants, plant biostimulants, and their influence on nutrient use efficiency, with particular focus on nitrogen use efficiency. And the last chapter deals with the combining plant biostimulants and the precision agriculture, which is very, very important for open field crops. Finally, I would like to thank several colleagues in the University of Naples, but especially the network of publication, Professor Colla from the University of Tuscia, Viterbo, Marius Chiriaco for the quality part, Luigi Lucini for the metabolomics and omics part, and also Paolo Bonini for all their supports. Hope, and this is all. Thank you very much.
Thanks, Prof. Rouphael, for this insightful presentation, which was rich of case studies and examples. I would go on with several questions we have received, if you are ready. So yes. The first one, thanks, Prof. Rouphael, for the very comprehensive presentation and for explaining the mechanism behind the biostimulant activity. You presented several research results dealing with the vegetal-derived biostimulants, but in the market we can find many animal-derived biostimulants. Can we expect the same crop response using animal-derived biostimulants instead of vegetal-derived biostimulants?
Yes, I will start from here. I don't know if my microphone is on.
Yes.
Okay, but I'm not able to find the platform. Yes. Look, yes, very nice question. Protein hydrolysate, when we talk about protein hydrolysate from a market point of view, he's right. The protein hydrolysate coming from animal origin are much more used than the vegetal-based protein hydrolysate. But as I said before, the difference, the protein hydrolysate coming from animal origin can give also excellent results. But as you know, protein hydrolysate coming from vegetal legume-derived protein hydrolysate can be applied, can be used in organic farming, while the animal protein hydrolysate is not allowed in organic farming. Another important issue that the frequent application, for instance, of protein hydrolysate coming from vegetal origin, we can apply, we can perform several applications without any toxic effects. While for animal application, we should be aware about the dose and the number of applications. And a third reason, very important, in the frame of circular economy, the protein hydrolysate from vegetal origin is growing very fast, especially because it's coming from a vegetal matrix, which is very important in the concept of the circular economy and farm to fork. But also protein hydrolysate is widely used all over the world. But for organic cultivation, only protein hydrolysate from vegetal origin are allowed.
Okay, thank you very much. And concerning the use of microbial and non-microbial biostimulants to reduce abiotic stress, such as alkalinity on vegetable crops, can you provide more information about the best application strategy for reducing such type of stress?
Yes. Why we are talking about microbial and non-microbial? Sometimes even as a concept of synergistic biostimulant action, it is not only to combine microbial and non-microbial. Sometimes we have a lot of product. We have seaweed extract and protein hydrolysate. The strategy is the following. We want to see if we have a synergistic effect. What does this mean? A synergistic effect, it means that the effect of this combined, of this product containing these two matrices is higher than the sum of the positive effect of these two biostimulants taken separately. What does this mean from a physiological point of view? This does mean that this product containing these two substances can trigger different pathway and can have an additional positive effect than these two biostimulants taken separately. Basically, why microbial and non-microbial biostimulants can be mixed and they can give a good result. Basically, because there is the logic that microbial biostimulants have a long-term effect, while the substances like seaweed extracts, humic substances, protein hydrolysate have a shorter effect. So combining these two categories, microbial and non-microbial, could probably trigger and boost the crop performance in a higher degree. Concerning the question about how and when we can apply, this is a very interesting question. And I mentioned this concept during my conclusions. So when we should apply biostimulants? Before stress, during stress, or after stress? When we are dealing with abiotic stress, it's very difficult for a technician to see that our plants are suffering from drought stress or nutrient stress. Because contrarily to biotic stress, we can see if the pathogens of pests attacks our plant. While in the case of abiotic stress, we cannot see, especially at the beginning of the stress, during the preliminary stage of the stress, the plants are doing a fitness course. So in this case, it is recommended to apply biostimulants directly after transplanting. So as a primer, so before the stress, the abiotic stress appears, so the plant is sensing, in the memory of the plant, it has some like primer in order to start the defense system. So my recommendation is that biostimulant, especially for abiotic stress, should be used before stress, just after transplanting as primer, and not during or as recovery. But even in this case, we don't have a lot of scientific evidences that support our hypothesis, because most of the scientific literature is based on experiment where the biostimulants were applied in the first period after transplanting, or directly or after 1 month of transplanting. It's very interesting to run experiment with to see if we apply biostimulants, microbial and non-microbial, during the stress, and why not as recovery after the stress.
Okay, thank you very much. Just a couple of other questions before we stop. In your view, which way of biostimulant application is more effective in greenhouse tomato for alleviating abiotic stress, like spray or substrate drench? Is there a way which is most effective?
Okay, basically, for sure, applying at root level, it's more efficient, and so the plant can, the root can translocate these signaling molecules and start triggering the primary and secondary metabolism. But from a practical and economical point of view, when we apply biostimulants, 80% of the application are done basically by foliar application. Concerning, as I said before, we cannot say these biostimulants perform better than another one. As I report during my conclusions, we should select the best combination. So biostimulant, the species or the cultivars tested, and the environmental agriculture practices applied. And one more time, because he asked when it is more effective to apply biostimulants directly after transplanting as primer.
Okay, perfect. And last question. As plants face a multitude of abiotic stresses in field condition, how significant is to use only one abiotic stress in the greenhouse experiment, predicting the efficacy of biostimulants in the field condition? Did you check the persistence of microbials in the field?
Yes, he's talking. He's saying in the field, basically, we have combined or multiple stress, right? While in the greenhouse, we are testing one type of stress, right?
Yes, exactly.
Yes, this is a question of $1 million. We should give them $1 million. This is a very, very interesting question. Yes, basically, you are right. Because when, for instance, when we are testing some abiotic stress, let's say drought in greenhouse, in the open field, we don't have drought alone. Basically, drought is combined with high temperature. And based on the scientific literature, I want to reply and to say that the effect of combined, the negative or the negative effect of combined stress is higher than the sum of the two stresses taken together. This is very important. Unfortunately, only in the last years, we start to run experiments dealing with multiple and combined stress. This is very important because under open field conditions, we don't have one abiotic stress. We have, for example, for salinity, most of the saline soil are also alkaline. As we know, for example, for salinity, we have the problem of the sodium and chloride toxic effect. But in saline soil, probably we have high pH, and under high pH, alkaline soil, we have another type of problem. We have the problem of the solubilization of phosphorus. We have the problem of chlorosis due to deficiency in iron. So it is very important. This is really a question of $1 million. There is a lack of information and strong data about the combined abiotic stress and the effectiveness of microbial and non-microbial biostimulants to mitigate the combined multiple abiotic stress under open field conditions. And another issue, very, very important, Under open field crops, we don't have only multiple abiotic strains. We have also combined biotic and abiotic, which complicate additionally the situation. We will be back in another webinar in 2-3 years, hopefully with some additional data.
Great, thanks for this answer. So we'll see you with additional data on this. So I would like to thank everybody for your time with us today. So if you still have questions, you can submit them on the biostimulant.com website, and our scientific committee will get back to you with the answer. And also, I would like to remind you that during this week, the record of this webinar will be available on the biostimulant.com website and on our digital platforms. So have a nice day, everybody, and see you on the next webinar. Bye.